Good morning, everyone. We are from the CityU iGEM team, or Very Cure, and our project will be mainly focusing on a new class of genetically engineered varicolin bioactive compounds. You might not understand what that is right now, but you will as we progress through the PPT. So this is what we'll be covering today, the problem that we'll be addressing, the possible solutions that we are proposing, the engineering that we're going to follow, as well as some contact information if you want to contact us later. Yeah. So let's start.
So let me introduce what problem the world are facing. But before we will figure out what problem our society is facing, let me introduce some background information. First, health is a fundamental for a good quality of life. And being healthy, being free from illness or injuries, will direct our capacity to enjoy our life. So, a population health can be measured by life expectancy, which measured how many years on average a person is expected to live based on current age and set specific death rate. Global life expectancy has increased drastically over the last couple of centuries, and with the advance of medicine, science, and in 2019, the world life expectancy was about 70 years, and compared to early 90s, was just only about 30 years. And this graph shows that that rising in healthcare expenditure will higher the life expectancy. But however, despite the increase in global life expectancy, there are still huge difference between the developed countries and developing countries. And the graph there shows that there's still huge difference between those countries. People live in South African countries have a life expectancy of less than 60 years. But compared to those developed countries, such as Japan, they have about 80 years. The reason is that the developing country are difficult to afford the drugs, and the people there it is hard to access the medicine. And the second problem that our world is facing, although major advance in medical science, the average life expectancy increased globally, but the number and type of human disease has been also increased across the world, which caused the demand of effective microbial and immunostimulatory drugs has increased, but the traditional drug discovery process is still really expensive, complicated, and time-consuming, which caused a problem on the supply of drugs. So for the next part will be the proposed solution for the above problem.
Yeah, so now that we've explained the problems that we'll be addressing to you, we'll be covering the possible solutions that we're proposing. But before we get into the nitty-gritty details, I'll explain an important part of our project, which is natural products. So as a definition of natural products, you can see are a set of small molecules which are derived from the environment and any life forms. Basically, any chemicals, compounds which are derived from nature are called natural compounds. And the sources of these are written right there from microorganisms, plants, animals, and fungi are a major source of natural products, which we will be capitalizing on. So as you can see from the graph made available, about 5% of the drugs that have been discovered in the last 40 years are from natural products, and 31% are from synthetic derivatives of natural products. So collectively, this forms about 40% of the drugs today, that is 40% of our arsenal against pathogens are derived or directly from natural products. Next. So why is this so? Why are natural products so important? So firstly, they have a high molecular mass. They have great molecular rigidity. They have a high hydrophilicity, and they also support a complex structure. So, sorry. So the high molecular mass enables us to employ the EPM concept, which is enhanced permeability and retention. We have molecular rigidity, which tackles protein-protein interactions, and hydrophilicities for easier drug delivery, and so on. Next one. Thank you. So for some of you, these are a few examples of natural products that exist in pharmaceuticals today. So you can see we have antibacterials, we have statins, we have painkillers, we have anti-cancer agents, and many of these chemicals today would probably be very familiar to most of you here. And one big example of natural products in drug discovery is artemisinin. Artemisinin is an anti-malarial drug which was discovered quite some time ago, but was awarded the Nobel Prize for Discovery and Development quite recently in 2015 because of research work that enabled it to provide its compounds, its chemicals in a lot faster and a lot more cost-effective way. So just to elaborate further, this is the current procedure that drug discovery follows today. So we have the extraction of the natural products from, say, a bacteria or a fungi. We have the identification of a crude extract. And mind you that this extraction process is just a mixture of all sorts of chemicals, so you don't know what's inside. And the identification process will identify maybe say one or two of these chemical products which have some pharmacological activity, but you don't know what. And then you go through this long-winded process of trying to divide and test again and again. It's a very long process. So this is what the current procedure looks like. And next. So what our project will be focusing on will be is a targeted discovery and not just look for what's there. We're going to be generating novel bioactive varicolin analogs using synthetic biology. So varicolin is a fungal sister terpenoid. Don't be scared by that term. A sister terpenoid is just a very big carbon compound, so it has 25 carbon atoms, and that's why it's called a sister terpenoid. And it was originally isolated from this fungus called Aspergillus varicola. And it was shown to possess very strong therapeutic activities from anti-cancer properties and antimicrobial properties. And we're going to be working on deriving more derivatives from this. So to summarize our project aims, we'll be focusing on one, to create novel varicolin analogs or derivatives from an Aspergillus fungal host. We'll be using a very well-known fungus called Aspergillus oryzae. And we'll be using something called P450 genes derived from several different other fungi for this process, which we will explain ahead in the PPT. And we will identify and then characterize these novel varicolin analogs. Basically, we just want to identify whether they are varicolin analogs or not by using GCMS, HPLC, as mentioned by the HKUST team, and even NMR spectroscopy. Furthermore, we want to, once we have detected these varicolin analogs, we want to test for their properties. We want to make sure that they have the antimicrobial and anti-cancer properties that we want. So we'll be testing through bioassays, MTT assays, through cell line testing and whatnot. And yeah, this concludes my part. So now, next up, Daisy will be talking about the engineering that will be following the project.
Before talking about the synthesis of varicolin, first we have to know what cytochrome P450 enzymes are. They are a family of monocygenase enzymes encoded by P450 genes. They play important roles in homosynthesis and metabolism and can function in drug metabolism. Here are some examples of cytochrome P450 diversification. By adding different P450 enzymes, different products can be formed from the intermediate. Now let's move on to the overview of biosynthetic process of varicolin. It starts with the condensation reaction of dimethylallyl pyrophosphate and 4-isopentanium pyrophosphate. DMAPP is the starting compound, while IPP serves as a building block that combines with DMAPP to give chain elongation. By using the enzyme terpene cyclase, the GFPP is then cyclized to form a compound with a 5865 ring. This structure can be further converted to ferrocholine by the introduction of 2 oxygen atoms using P450 gene. Different P450 genes can also be introduced to the intermediates to give new ferrocholine derivatives, which may have different biological activities. In this project, 23 different P450 genes are tested to see if ferrocholine derivatives can be synthesized. For the selection of the 23 P450 genes, we expected that the fungus Aspergillus aculeatus contains the gene that is responsible for the biosynthesis of ferrocholine. Then a program C-Blaster can be used to search for the P450 genes from other fungi and compare the similarity with the target gene. For the genes that have higher or lower similarity with the target gene, we will select some of them for further experiments. After checking the similarity, we can do the synthesis of ferrocholine derivatives according to this flowchart. The gDNA is first extracted from different fungi, and the plasmids are constructed with P450 genes, followed by transformation of its recombinant plasmid into a fungal host and selected the transformants. Then metabolite production, extraction, and analysis will be done, and finally, we will do assay of products to examine their biological activities. Firstly, we need to do genomic DNA extraction for P450 gene amplification using PCR. The first step is lysis of fungi. We do micropipetting to crush the fungal mycelia in lysis buffer. Then we add extraction solutions and vortex to mix them together. The second step is precipitation. We centrifuge the sample and collect the layer which contains the gDNA. After that, isopropanol is added to precipitate the DNA from the solution. The last step is elution. After centrifugation of the sample, we add ethanol to wash the pellet. Finally, the pellet is dried and dissolved in TE buffer. At the same time, we need to design suitable primers for fungi using bioinformatics tools. Then the P450 genes can be amplified in the process of PCR. In plasmid transformation, we clone P450 genes into E. coli DH5 alpha by homologous recombination. First of all, P450 genes are ligated into plasmid, and then it is inserted into E. coli by chemical transformation. Finally, transformants are plated and selected on ampicillin plates. After we have amplified the plasmid with P450 gene in bacteria, we move this PTX3 P450 recombinant plasmid into a fungus called Aspergillus oryzae, and this step is called PEG-mediated protoplast transformation. The mechanism of transformation includes 2 steps. The first step is CRISPR-Cas9 genome editing. Guide RNA and Cas9 can attach to each other, and guide RNA will attach to the target gene sequence in the protoplast, leading Cas9 to the target gene sequence and cutting it, causing DNA lesions. After that, the lesion of protoplast DNA will be replaced by the recombinant bacterial plasmid, which contains the P450 gene. As this recombinant bacterial plasmid is homologous, with the 2 ends of it being identical to the DNA lesion of the protoplast, it can integrate into the protoplast genome automatically. Finally, we can obtain the transformed fungi with P450 gene, which is shown in this picture. After transforming the P450 genes into fungal hosts, how can we make sure the transformation is successful or not? This is the purpose of the selection method. Our selection method is based on the auxotrophic mutation happening during the growth of Aspergillus oryzae host. In our host, There are 3 orthotrophic strains, and they need adenine, arginine, and methionine to provide corresponding nutrients for hosts to survive. TC gene-containing plasmid and P450 gene-containing plasmid provide the genes for the fungal host to synthesize its own adenine and arginine. If P450 gene-containing plasmid transformation is unsuccessful, our host, Aspergillus oryzae, will not grow. This is an example of the selection results of one P450 gene transformation. The picture on the left is our products after P450 gene transformation. After selection, the fungus in minimal medium agar plate showed the result of successful transformation. Then the metabolites produced by successful fungal transformants are analyzed by GCMS and animal spectroscopy. Their biological activity will also be tested.
If anyone wants to know more about our project, they can scan this QR code and fill in our public opinion survey, which has a bit more information about our project and a few questions for you to answer. And I'll just leave this up for like another 10 seconds. So yeah, it's like 10, 9, 8, 7, 6, 5, 4, 3, 2, and 1. Okay. Yeah, so thank you for anyone that scanned the code, and yeah, thank you. We're open to questions, and this is our Instagram page and our email ID. So any questions?
Okay, I had a question, like, just to clarify a bit, can you hear me?
Yeah.
Are you basically taking the P450 gene from oryzil, the main fungi, and transforming it into different strains that you think are closely related?
No, just to clarify, we know that we have an initial strain, right? Our source strain is Aspergillus aculeatus. We know that produces varicolin, right? So we've tested, there's this genome database called Blaster, and we're using it's called BLAST, and we're using C-Blaster to find, we found about 23 fungi which have a similar gene cluster to the one that we want, right? So we have 23 P450s from different fungi that we know if are very similar to the one we're using. So we take these 23 P450 samples that we've found from C-Blaster, and we edit them into the Aspergillus oryzae, which is the host sample we're working with. And once they're edited into the target, like the host that we're using, we let it grow, and once it's successfully transformed and we select it, it'll produce chemicals in the agar plate that we're growing it in. So then we extract those the chemicals that it's produced and move on to the testing phase.
Right.
So,
Makes sense. Okay, thanks.
Do you mind explaining what are the rationales behind using a fungal host to produce your target compound instead of using more common, industrially used species?
yeah. So basically, our entire research project was inspired by another research project which produces varicolin from a professor. His name is Yudai Matsuda, and he's working on varicolin synthesis. And varicolin was inherently produced by a fungus called Aspergillus varicola. So when we're testing to see if we can produce varicolin in other compounds, it has to be a fungus because the source material, like mentioned in the PPT, DMAP, IPP, P450, all of which are present in fungi. So we kind of had to use fungus to continue with the seeds.
But then in the long run, if
So far, we haven't reached the point where we're thinking about commercialization, but right now, the entire process to produce the fungi and detect the chemicals takes about 4 days. So right now, we would say that it's not quite there yet, but we're working on it because we still need to. We're still in the testing phase, so we're not. We have produced about. We had about 23 P450s, out of which we detected 12 successfully transformed, and now we're in the testing phase. So we've extracted the chemicals, and we want to test if they have the activities that we want them to have. And once we've finished the testing phase, only can we move ahead and think about shortening the prep time and commercialization and everything. I hope that answers your question. Thank you.
How is the growth rate of the fungi that you use? Like, how long does it take to actually see the clones, if it's successful, if it's not?
Yeah, I just mentioned 4 days.
4 days.
Yeah.
So you have to wait that long?
Yeah.
Okay.
I have two questions. The first one is, would you mind to explain a bit on how vertically works in antimicrobial and anti-cancer functions? Like, are there any specificity on their mechanisms? Like, what types of microbes is acting on? And the second question is, I might need a little bit clarification. Like, you are extracting DNA from the genome DNA, right?
No.
No. Okay. So yeah, then you can answer.
Yeah. First, the first question that you have, varicolin. Basically, I don't remember the entire. Like, we did a bit of literature review, so I don't remember the entire thing right now. But based on what we the review that like the literature that we read and our contact with Professor Matsuda, we've seen that in Professor Matsuda's lab, varicolin has we have assay tests where you have like an agar plate. You put in a bit of varicolin inside and you let it grow with, say, bacteria or fungi. That's how antibacterial assays work, right? So it was shown to have a ring of no growth around where varicolin was placed. So the inference was that it has antibacterial properties, and the anti-cancer properties is what we want to test. We are not 100% sure about that. We've done a little bit of literature review saying that varicolin, because tumor cells grow at a very fast rate, right? So when tumor cells are growing, they take in a lot of the macromolecules that are in the bloodstream. So in certain other tests, varicolin was shown to have high concentration, varicolin was shown to enter the tumor cells, say in breast cancer cells. I mean, it was breast cancer cells, not say, my bad. Breast cancer cells, they took up varicolin and the growth was a bit stunted. I'm sorry, I'm not aware of the exact details and the numbers, but yeah. And also the second question that you had we're not extracting the fungal chromosome from the cell. We're just dissolving the cell wall of the fungus to have a protoplast, and then we want to use PEG mediated transformation process to have the Cas9, the protein, the gDNA, everything, enter the protoplast through this. So it'll be happening inside the cell protoplast, the entire process.
Because I've seen in your outline you mentioned you precipitate the gDNA from the fungus.
That's other fungus, where the one we are extracting P450 from. Yeah.
you mind explaining a bit more about how, like, what's the activity of what the very hot. Like, what is it actually targeting? What is it actually doing cancer cells in bacteria?
So basically, as I mentioned before, right? Right now, not we're not completely aware of how it works because varicolin is has only been recently been synthesized in the lab, right? I mentioned Professor Matsura. So and he's also working on the derivative research. But what we'll figure out the exact process of how it works as we move through the testing phase. We haven't reached the testing phase yet. So.
And how do you plan on, say, for cancer cells? How do you plan on deducing whether the drugs are effective? How do you screen the effectiveness for the drugs?
So, yeah. So basically, so far for the anti-cancer cell research, we have two methods in mind so far. One is the MTT assay test, and the other one is using actual cell line. So the MTT assay that we'll be working on, based on what I'm aware of, one of our advisors suggested it. Once you have once you add varicolin into the MTT, in like a basic sense, once you add varicolin into the MTT assay, you will see a color change in the assay, which basically deduces the if it has anti-cancer properties or not. Because I'm not aware of the exact details of the MTT assay part. I'm not involved in that research field. If you want more information. But the other cell line testing that we have is for, we're going to be using Mabrigand fibrocystic disease cells. We're in talks with HKUST as a collaboration event to provide those cells. And we want to test if the chemicals that we've extracted have any effect, say stunted growth or any effect at all on those cells. So that's how and anyway, that's how research works, right? You trial and error. So we want to see if it has any effect. If it doesn't, we'll probably move on to a different focus.
Thank you very much, CityU iGEM team.
Yeah, thank you everyone.
Okay, so right now we're gonna have a 15-minute break so you guys can go to the washroom.